Hello everyone, and we will see the very important experiment. Just I have discussed you all the theoretical parts related to the significance, theory, and what are the usefulness of this corresponding experiment. That is nothing but dissolved oxygen, and it is newly introduced into the syllabus. Previously, dissolved oxygen experiment it is limited for the wastewater, but now due to realizing the depthness or due to realizing the importance of this corresponding experiment, syllabus has rescheduled, and in that syllabus they have introduced this new experiment that is nothing but the dissolved oxygen. Very important experiment. Each and every engineer, civil engineer particularly, who are working in the field of water supply and wastewater engineering cannot do any analysis without the reference of this very important characteristic, that is nothing but the dissolved oxygen. Theory I explained, definition I have explained. Now it's time to learn what are the reagents required and what is the procedure before actually starting the demonstration of the corresponding experiment, dissolved oxygen. Let us see first what are the reagents required. I have written here the reagents which are required for this experiment. First reagent required is manganese sulfate, that is MnSO4. This reagent or chemical we have prepared in our laboratory. Then next reagent, second reagent required, that is alkali iodide azide reagent, that is also very important second reagent which is required, or chemical. This is also we have prepared in our lab. Then third is nothing but sodium thiosulfate, Na2S2O3, and this sodium thiosulfate has its significance in this particular experiment is that in this particular experiment we will going to use sodium thiosulfate as an important titrant. And fourth number chemical is nothing but concentrated H2SO4, and in this process H2SO4 acts as a catalyst. H2SO4 acts as a catalyst which increases the speed of reaction, or we are knowing catalysts are nothing but what? These are nothing but the simple chemical or material which increases the rate of reaction and which increases the speed of reaction. And mostly chemical engineer, or in this term catalyst is mostly in the chemistry or chemical engineering term. But this experiment we have used this concentrated H2SO4 and its concentration is 98%. Highly concentrated H2SO4 is required in order to perform this experiment. Then fifth type of the chemical, it is not chemical, it is simply the indicator, and that indicator gives the notation, gives the information about the color change has occurred and your titration has reached a end point, and this is process of titration or volumetric analysis. And particularly talking about the procedure, just we have seen the corresponding meaning of the corresponding reagents which are required. Now we will see particularly the procedure. Just a minute. Now we will see particularly the procedure which is required for this test. Now we will see the procedure required for this test. Just a minute. I have written here the procedure, and particularly talking about the procedure, first case is nothing but, I hope that all of you are visible this procedure. Step number 1 is determine the presence of oxygen. As I have discussed with you in our theoretical session, and in that theoretical session I have discussed with you that this is which process? This is reaction process in which all the chemicals are used, and that chemical will going to react with each other, and finally after reaction of chemicals with each other they are going to liberate the iodine, and this is nothing but reaction releasing iodine. म्हणजे ज्या रिएक्शन आपण करणार आहे आणि लास्ट ज्यावेळी रिएक्शन होणार आहे त्या रिएक्शन मध्ये आयोडीन लिबरेट होणार आहे. कशातला आयोडीन लिबरेट होणार आहे? इथे जो मी सांगितलं अल्कली आयोडाइड अझाइड रिएजंट, त्यातला आयोडाइड कन्वर्ट होणार आहे आयोडीन मध्ये, आणि जो त्यावेळी लिबरेट होईल त्यावेळी आपल्याला काय मिळणार आहे? वी विल नियर टू द एंड पॉइंट, अँड दिस इज रिएक्शन रिलीजिंग द आयोडीन, and that iodine we have to liberate, iodide we have to convert into iodine. How that it will going to convert into iodine, we will see step by step. First we will take step number 1. Now step number 1 is nothing but determine presence of oxygen. First we have to determine the presence of oxygen. Oxygen is present or oxygen is absent. If oxygen is absent, there is no any need to perform the titration, there is no any need to add the indicator, because dissolved oxygen is not present. How we will able to perform the titration? Hence this is nothing but the very important step. If oxygen is absent, then waste of doing this all the processes. I think total step number 5 we are having all the processes. If oxygen is absent, then you are not having an authority. If you have do any analysis, you are not going to get any result related to this particular experiment, that is nothing but the dissolved oxygen. Here in the step number 1, first we will determine oxygen is present or oxygen is absent. Here I have written that add manganese sulfate and alkali iodide azide reagent. Means first step we will going to add the manganese sulfate, the chemical just I have told to you, and that we are going to add in this corresponding bottle. Sorry, one bottle that I will show to you. This bottle is known as the dissolved oxygen bottle. In this dissolved oxygen bottle, we will going to perform this experiment, and all the chemical reaction you are going to observe in this corresponding bottle. That bottle we will going to use for our analysis. Add manganese sulfate and alkali iodide azide reagent. We will going to add all these reagents which are mentioned in the step number 1. MnSO4 plus KOH, that is manganese sulfate, plus alkali iodide azide reagent in this corresponding bottle, that is DO bottle. And then at this stage we will able to estimate that oxygen is present or oxygen is absent. And when both these two are added in this DO bottle, reaction will going to occur and formation of manganese hydroxide will going to occur. Here I have written manganese hydroxide will going to generate. When manganese hydroxide will going to generate, you are going to get the white color. And when you are going to get the white color of solution in this DO bottle, it is an indication that dissolved oxygen is not present. Dissolved oxygen is nothing but absent. You have to stop the process, you have to stop the experiment, because dissolved oxygen is absent. There is no any use of doing the further analysis. When dissolved oxygen is present, then what will going to happen? When dissolved oxygen is present, then this manganese hydroxide which is formed in this corresponding reaction, that is going to react with this oxygen which is in the dissolved state. Here I have written the second reaction. It is occurring when dissolved oxygen is present, that is manganese hydroxide will going to react with the oxygen which is present, and formation of manganese oxide O2 plus H2O, that is brown manganese dioxide, that is now going to be formed. And when it will going to form? It will going to form when oxygen is present in your corresponding sample of water. And when oxygen is present in your corresponding sample of water, brown color is developed. When brown color is developed, then it is an indication that you have to perform the corresponding titration. Dissolved oxygen is 100% present in the sample. Just a minute, I will show you one sample. This is nothing but one sample. In this sample, these two chemicals, as a analysis rough experiment, I have added these two chemical in this beaker, but no, you have to add this in the dissolved bottle. Just for your understanding, I have added in this beaker. You are able to observe that brown color is developed. When such brown color is developed, that is manganese hydroxide dioxide is generated, MnOH2, and that manganese dioxide is indication, that brown color is an indication of what oxygen is present in your water. Then your step number 1 is completed. You have to follow the step number 2. If white color is generated, you have to stop the titration. There is no need to perform the further analysis. Now you will see the step number 2, that is oxidize iodide to form iodine. Okay, how we have to carry out the oxidation? Because if we keep the sample steady for one day, 2 day, 5 day, speed of oxidation is very low. Speed of oxidation is very low is nothing but what? Oxidation is nothing but the reaction of liberation of combination with oxygen and liberation of certain gases. When such process of oxidation is required at a faster rate, we require the help of the catalyst, and that help of the catalyst we require, that catalyst is nothing but, in this experiment, that is nothing but the concentrated H2SO4 that we will going to add in this step. Here you are able to see that the manganese hydroxide brown color is going to form, that is going to react with the H2SO4, and catalyst we have added, this is Ki, that is nothing but already present iodide. in your corresponding sample. And manganese sulfate, K2SO4, potassium sulfate plus H2O plus, in this case at the last you are able to observe that this is I2, this is nothing but what, liberation of iodine has occurred at this stage when you have added the concentrated H2SO4 into the sample. When you have added concentrated H2SO4 into the sample at this stage, liberation of iodine has occurred, and now it is an indication that we are nearer to the end point. We have to perform the titration. Okay, step number 2 is completed. Then step number 3 is titration. Titrate the liberated iodine with the standard sodium thiosulfate solution that we have prepared in our laboratory, and that solution we will going to use it as a titrant, and we will going to perform the titration. And step number 4, directly titration without the help of the indicator is not possible, hence we are going to use the indicator, that is starch indicator we will going to use. And color changes, when we add starch indicator, color changes to blue, first yellow to blue and blue to colorless is nothing but the end point of this experiment. And step number 5 is nothing but this thing which I have discussed, just continue the titration till solution becomes colorless, and when your solution becomes colorless, that is nothing but the end point of this corresponding experiment. Iodine has liberated, we have treated that, we have titrated that liberated iodine with the sodium thiosulfate, and at this stage the end point which we achieve, that is directly exactly proportional to the amount of dissolved oxygen present in the sample. There is nothing but procedure of this experiment, very interesting experiment, as well as reagents required we have seen, and one of the very important as well as interesting procedure. Why I am talking you that this is interesting, after doing the demonstration of this corresponding experiment, you able to know that why this experiment is important and why its procedure is quite interesting. We will see the actual demonstration of this video. Thank you for listening the procedure, and we have just studied the reagents. Let's move towards the demonstration. We will see the demonstration of this corresponding experiment, dissolved oxygen.